Good afternoon. My name is Gomozova Tatьяna Sергеевна. I'm a student of Приволжский Research Medical University, and today I'm going to tell you about fluorescence lifetime imaging microscopy as a highly sensitive and specific method for diagnosing atypical cell division and oncological process. It's important to understand that the altered metabolism of tumor cells is directly related to the features of cancer that distinguish it from healthy cells. Such features include a pathologically high level of proliferative activity, the presence of adaptations not characteristic of a normal cell, changes in metabolic status, activation of oncogenes, and loss of tumor suppressions. As a result, anabolic processes increase in cancer cells, changes in bioenergetic occurs, and the redox balance changes. Cells need energy to maintain homeostasis. All processes of cell vital activity, such as the creation of concentration gradients, cytoskeleton movement, DNA repair, transcription, translation, and vesicle transport, are energy dependent. It's known that the energy metabolism of normal cells differs significantly from the metabolism of cells in certain diseases. Therefore, the metabolic status can serve as an indicator of diagnosis as well as visualization of the response of the treatment of pathological processes. Due to the wide prevalence of oncological diseases, their assessment of the metabolic phenotype of tumor cells is especially relevant. The most important pathways of the energy metabolism of the cell, such as glycolysis and oxidative phosphorylation, are associated with the process of cellular respiration and ATP synthesis. NADH and PET effectors act as electron donors and acceptors in ATP formation reactions, which are present in the cell is oxidized NAD plus, PET, or reduced form NADH and PETN2. Unlike normal cells, in which oxidative phosphorylation is the main method of ATP synthesis, most cancer cells have an increased level of glycolysis. During glycolysis, a glucose molecule breaks down into two pyruvate molecules, while two NAD plus molecules are reduced to two NADH molecules, and two ATP molecules are formed in substrate phosphorylation reaction. Firstly, necessary monomers are formed as a result of glycolysis for the synthesis of nucleic acids, proteins, and lipids. Secondly, during glycolysis, the formation of free radicals is reduced, which reduce the level of gene toxic damage in the cell and helps to avoid apoptosis. Thirdly, the product of glycolysis is lactate, which is transported into the intercellular space. It's known that acid environment promotes tumor invasion and metastasis. The study of cell metabolism by the method of time-resolved fluorescent imaging of limb is based on the fact that the fluorescence lifetime of NADH and PET effectors differs significantly depending on whether they are in a free state or are bound to proteins. Fluorescent imaging with time imaging makes it possible to study metabolic effectors in living cells by measuring the average time during which the molecule was in an excited state, rather than the actual intensity of fluorescence. The lifetime may be affected by pH, temperature, iron, oxygen, concentration, binding to other molecules, or chromophore conformation. Since NADH and PET are the only electron carriers capable to fluorescence, the duration of their fluorescence intensity can serve as a parameter for assessing the metabolic status of cells and tissues. Since the bank of NADH is formed during glycolysis, the PET is formed during oxidative phosphorylation. The rate makes it possible to understand the way energy is produced in the cell. For example, an increase in an NADH concentration is a characteristic feature of tumor cells. The ratio of oxidized electron carriers to reduced ones is called the redox ratio. Today, the redox ratio indicator is used to assess the metabolic status of tumor cells in vitro, ex vivo tissue, and of course, in vivo tumors. Due to its non-invasiveness, highly sensitivity, and the absence of the need for the introduction of exogenous days, the FLIM method has become an indispensable tool for visualizing tumor metabolism. Recently, the application of the FLIM method in clinical oncology has been of particular interest. The analysis of cofactors in human tumors is carried out mainly in biopsy samples. In the work on ex vivo human breast tumor samples, it was revealed that tumor cells with desmoplastic reaction had a shorter lifetime of radiation-fed fluorescence compared to tumor cells in which this reaction was not observed. Thus, the FLIM method is a specific and sensitive way to diagnose any pathological process of whose cellular metabolism differs from normal. With the help of the FLIM method, it's possible to diagnose the development of the oncological problems at its early stage and, of course, begin early treatment. That's all I want to say. Thank you for your attention.